Hi everyone, we're group 6 from discussion 4. Our presentation is about examining the consequences of mismatch repair in Pseudomonas and aeruginosa. Our group members are Wayman Yu, Joseph Fonseca, Kayla Chacon, and Xochitl Estrada. First, we're going to take a look at paper 2, which is examining the consequences of a defective mismatch repair in Pseudomonas and aeruginosa. The main point of this paper was DFBM, which stands for Distribution of Fitness Effects of Beneficial Mutations. This point has two key features, one being the number of beneficial mutations available for selection and the shape of the distribution. For any given mutation, the supply rate and rapid evolution of resistance will be when the distribution of fitness effects is towards mutation or when it is skewed towards mutation with a larger effect. Recent work within the allocated paper demonstrate that the molecular basis of resistance and antibiotics does play a key part in determining the DFBM under the selection for antibiotic resistance. The dosage of the antibiotic is also a key player within the paper. The authors state that low drug doses that only partially inhibit the growth of the sensitive bacteria and that there is a weak selection within that resistance. However, there's a low fitness cost that will have a higher fitness compared to the wild type allele presented. The paper also highlights the work focused on DFBM for spontaneous mutations and how antibiotic resistance is frequently gained through horizontal genes transfer. The dynamics of plasmid transfer depend on the population's demographic, which suggests that the fitness cost within plasmid-borne resistance is also a great contributor of the resistant plasmid in heterogeneous bacterial communities. Experimental evolution also allows us to study evolutionary practices in controlled environments like a laboratory. This comes with several advantages over studying adaptation to the natural population. One advantage of this paper is that the amount of time that could be saved that you could choose to examine microbes, since it does have a short generational time, and then use what we have learned to apply to a larger organism that has such a much longer generational time. Another advantage is that we can control nearly every aspect of the experiment, like environmental factors, example for temperature, and the genetic makeup of a subject. For example, genetically uniform and genetically diverse. Another advantage that we can do is whatever necessary to test our hypothesis without having to worry about ethical concerns, since we would be mostly working with microbes for experiments relating to those evolutionary processes. The distribution of fitness effects of beneficial mutations, DFBM, is a way to visualize and examine the proportion of genotypes in relation to fitness growth rate, which tells us that the genotypic variation within the population, which influences the amount within that population that could obtain resistant mutations. For example, we can examine organisms that are low in antibiotic concentration and high antibiotic concentration. Based on the DFBM, we can see that the low antibiotic concentration, there are wild types of bacterial cells that have the same fitness as those that have resistant mutations, while during high antibiotic concentrations, all the wild type cells are not fit, and those with the mutation have a higher fitness. Also, one last thing would be that the exponential DFBM relies on the wild type high fitness because it is needed in order for the beneficial mutations to correspond to the samples from the tails of the distribution.
first image on the top left demonstrates the experimental process by which scientists use to bring rise to antibiotic-resistant strains in an organism via antibiotic agars. In this experiment, they visualized how the resistant strains ended up developing independent colonies. The bottom part of the left image demonstrates how bacteria use mobile elements to intercede for horizontal genes transfer of potential advantageous genes. Due to mobile elements having tendency to occupy fitness costs to preserve effectiveness, these genetic mobile elements, compensatory adaptations are required to alleviate cost. The second image on the right displays different situations in which resistant mutations may arise in relationship with the diminishing proportion of genotypes and the fitness of wild-type sensitive genotypes.
The second paper we used is paper 11, titled Mutation, Recombination, and Incipient Speciation of Bacteria in the Laboratory. MMR stands for methyl-directed mismatch repair. It is a system that recognizes errors in DNA replication. Defective MMR leads to high mutation rates and could lead to antibiotic resistance if antibiotics are present in the environment. The effects of defective DNA MMR include high mutation rate and recombination rate because DNA is unchecked for errors and the proteins involved in regulating recombination are inhibited. The pairs of genotypes used in this experiment are functional MMR, MMR plus, and non-functional MMR, MMR minus. The experiment performed crosses between a donor and the MMR plus version of a given strain, simultaneously with crosses between the same donor and the MMR minus version. Results were measured by observing the ratio of recombination rates. The ratio of recombination rate measures the genetic barrier created by functional MMR. A higher ratio describes strong inhibition of recombination when MMR is functional. Strong inhibition of recombination tells us that there's more sequence divergence between the donor and recipient. The results of the experiment were that mutator lines diverged from the ancestor to an extent that was sufficient to create a barrier to recombination in the presence of MMR+. The results from this paper support that populations with defective MMR experience rapid genetic divergence and that sequence divergence does not block recombination if MMR is defective. However, if MMR becomes functional again, the cumulative sequence divergence becomes a barrier when it comes to recombination. DNA MMR is an important process for regulating the stability of a cell's genome. MMR proteins involved in the checkpoints of the cell cycle cannot do their job if DNA MMR is defective. DNA MMR checks for base-to-base mismatches and insertion or deletion mispairs in the recombination and replication of DNA. Defective MMR impairs the cell's ability to detect DNA damage and activate apoptosis if necessary. Defective MMR allows cells to have high mutation rates in their genomes. These mutations allow replicated cells to have increased chances of being resistant to antibiotics. The graph on the right represents different crosses and their log ratio recombination rates. A higher log ratio recombination rate value on the y-axis means that there is a stronger inhibition of genetic recombination due to functional MMR. The figure on the left shows the strain deviations A+3, A+2, A-2, and A-1. Mutated genotypes with defective MMR took over A-2 and A+3, while A+2 and A-1 still had functional MMR.
For our outside source that looks into how a defective DNA methyl-directed mismatch repair, or MMR systems, would affect the rate of antibiotic resistance acquisition. Bacteria employs various methods in order to maintain DNA replication fidelity. One such method is through the use of MMR systems, where it reduces the spontaneous mutation rates during replication. What the researchers found through this experiment was that in ceftazidime, avibactam, or CZA resistant isolates of Pseudomonas aeruginosa, there was a R656H substitution present on a conserved area in the ATPase domain of the MUT-S protein. This resulted in an inactivated MUT-S protein leading to a defective MMR, which caused an increase in spontaneous mutation rates. In vitro adaptive evolution experiments were then performed on the Pseudomonas aeruginosa strains that lacked MUT-S protein when in the presence of CZA. This showed that they obtained CZA resistance at a much faster rate than their wild-type counterparts. On the top image, you can see where the R656H is located on the MUT-S protein, which is shaded in light blue. On the bottom image, graph A, the y-axis represents the highest CZA concentration where growth was still detected, and the x-axis represents the number of passages that the sample has undergone in serial passaging. This graph tells us that the MMR deficient isolates shown here as MPA01 MUT-S minus still had growth even at really high CZA concentration when there was no growth at the same point for the wild type. On graph B, it tells us just how much passages it would take to obtain growth when with CZA present. And here it shows that MMR deficient isolate took less passages for it to obtain growth, which means that it obtained resistance to CZA at a much faster rate than the wild type. Given that mutation is what drives adaptive evolution, a defective MMR that is unable to remove DNA replication errors would accelerate the adaptive evolution of organisms through the increased mutation rates and genetic diversity. This would result in giving a bacterial population a faster rate of increasing its overall fitness when placed in a new environment, which would allow it to more quickly adapt. Defective MMR also contributes to speciation through the genetic divergence caused by the increased rate of evolution from the increased rate of mutations.
Through the study of clinical isolates of Pseudomonas aeruginosus, it has been determined that those that lack MUT-S protein results in the increase in spontaneous mutation rates using rifampin reversion assay and whole genome sequencing. And through the experimental evolution in vitro that was performed, it showed strains with a defective MMR from an inactive MUT-S protein resulted in a faster rate of antibiotic-resistant acquisition. When comparing the clinical isolates and the experimental isolates, we could determine the cause of the change in rate of resistant evolution was from the defective MUT-S protein. This also supports that not only does MMR play a role in speciation, but it also plays a role in antibiotic resistance. It contributes to speciation through the genetic divergence caused by the increased mutation and recombination rates. Genetic background of the organism affects the distribution of fitness effects of beneficial mutations by revealing the underlying biases of the number of different type of mutations that are present. In this case, it would be the overrepresentation of transversion mutations caused by MUT-S. An organism with an affected MMR shows high level of mutation and recombination, which leads to increased rate of adaptive evolution. Through this understanding of knowing what proteins are necessary for a normal functioning MMR, we are able to merge the concepts of prolonging protein function of non-antibiotic resistance so that recombination of new alleles may be halted and the activity of developing new antibiotic-resistant alleles is minimalized. This will pave the way for more efficient antibiotics with less selective pressure upon pathogens to rapidly mutate and become resistant.
Thank you for listening to our presentation.